All right, so it's my great pleasure to introduce Dr. Etienne Yergeau. He's from INRS with the University of Quebec network of universities, I guess. And so Etienne studies the microbiome, so that covers the outside and insides of plants and trees, crops, animals, ice, water, snow, what have you. He completed a master's degree focusing on fungal pathogens associated with asparagus. And then followed with a PhD looking at the impacts of climate change or global warming on Antarctic soil microorganisms. He now leads a team of very enthusiastic scientists who are making exciting discoveries in the field of plant microbiome interactions, and he's going to talk to us today about mainly about their modeling efforts, I think, in terms of understanding the plant microbiome, but how modeling and engineering plant and soil microbiomes is advancing in his research team.
Okay, thank you. Or maybe can I use this one? Yes. Because I might start walking. That's something that happens when I talk. So, yes, thank you for inviting me, and thank you for the introduction. And also, it was perfect with what you showed because it's going to save me a lot of explanation of a lot of my slides. So, yes, I'm an associate professor at the INRS. That's in my institute is nearby Montreal. So. And so that's what I'm planning to show, and this is very exciting for me because it's a brand new talk. So I made it especially for you guys to match with the digital agriculture team of the conference. So I'm not sure if I'm going to go through all of that, and I don't know who's keeping time, but please be very strict with me so I don't go and take time for the students in the flash talk. I wouldn't want to do that. So I'm going to talk about microbes as key players in ecosystem processes, also as microbiome as ideal predictor descriptors of ecosystem, and these two things together we think that makes them ideal also to do some modelings of different things. And I'm going to talk about our recent efforts to model the wheat grain quality and yields in Quebec, and then what can we do with these models afterward. Basically, I'm going to show two examples, and then maybe we have time to talk about microbial assisted agriculture. So I'm mainly going to focus my examples on nitrogen because there's a clear crisis in nitrogen, and there's a target to reduce nitrogen use, fertilizer, or at least the greenhouse gas emission due to the fertilizer by 30% by 2030. In the Netherlands, it's already it's way higher than that. It's 50% that you need to decrease, and I don't know if it's riots, but people are really angry about that because it's difficult to change so dramatically your way you produce your crop. But basically, nitrogen use need to decrease, and the efficiency needs to increase. And could microbe help? And here I showed a picture of what happens from fertilizer that is produced with the Haber-Bosch process. If you talk about 100%, you start with 100, then what end up in your crop is probably 47% of that, and a lot of that is lost here. And this is where microbes are very active. So here I'm showing a simplified view of the nitrogen cycle. So microbes are involved in transformation of the nitrogen in a soil. So there's two main groups I'm going to focus on today. The ammonia oxidizers, so they take ammonia and they bring it to nitrate in a two-step process. And the ammonia oxidizers are normally the limiting step for that. That is called nitrification. And then there's another group of microbes that will take the nitrate and remove it, so transform it into gaseous intermediate, so it's going to go out of the soil. So these are the two main groups we're going to focus on, but these are the main drivers of the change in nitrogen. And we're going to see later that what type of nitrogen is in the soil has an effect on how much energy the plant needs to spend to create protein, let's say. There's many other examples where microbes are key players. So, Bobby talked about a few of them, but there's the promotion of root growth, pathogen suppression, tolerance to abiotic stress, phosphorus solubilization. All this affects the plant phenotype. So there's and often there's maybe we can discuss that, but often microbes can be let's say not taking into account in some of the breeding effort because they can affect the phenotype, and they're also responsible for many processes in the soil, like I said, the nitrogen cycle. And at the same time, microbes are integrators of what's going on in your soil. So if you look at the abundance of a certain microbes relative to soil water content, you will see that this microbe, if it's too wet or too dry, it's not going to be there, and it's going to have an optimal abundance at a certain water, soil water content. So if this microbe is abundant in a certain soil, that means that this soil has a certain soil water content that is conducive to the life of this organism. And you can do that with all kind of different other indicators: temperature, nitrate, organic matter, pH. And this is for a single microbe. So you have thousands of species of microbes in each gram of soil. So our idea is that microbes integrate a lot of physicochemical characteristics in their abundance, and then if you have a community portrait, you can recapitulate a bit of the soil conditions, for instance. And not only of the current condition, but we also showed that it's also related to past conditions. So there's a signal in the microbial communities that is related to the legacy. So in that example here, this is an ordination, so each dot is a different sample, and the position of the dot is depending on the microbial community. So further apart are the dot, more different are the microbial community. So you can see there's a clear separation between the open and the closed symbols, and these are from two fields that are adjacent. It's here in Saskatchewan. It's near the Swift Current Research Center. So one of the fields was irrigated for over 40 years to test. It was a test for new genotypes. So there was one field that was irrigated, the other not, and it was to compare how the genotype were doing in these conditions. But by doing that, they created a legacy into microbial communities, or the soil are typically the same, but the microbes are completely different. So microbes are indicator, or they can give you an indication of what happened in the past. And also they're influenced by the biotic interaction. So if you look at for instance, my curve here of the abundance of microbe for a certain parameter, then this will be influenced by the interaction with other microbes. So it might squish it. So let's say the microbe will have a lower range, or it can push it toward different values. So all this makes us think that microbes are good indicators. And also what's very interesting with microbes is you can take a gram of soil or gram of anything, in fact a plant, and you can crush that, extract DNA and sequence it, and you're going to get the entire genetic information of all the microbes that were present in this sample. So we get thousands of different genomes in a single gram of soil, and we think that these genomes, they contain information about the preference of the microbes in terms of physicochemical characteristic, also of the legacy, and also of the biotic interaction. So they're kind of recapitulators of all these things. And so our idea is that microbes are at the center of that. So if you talk about all these things, climatic condition, environmental characteristic, past and present, it's all integrated in the microbiome. And then if you can use this microbiome with some statistical learning tool, you can predict different ecosystem processes. So that's what we call the predictive power of the microbiome. And this is a paper we it should come out online soon, that we just make the case I just made you probably less confused and easier to follow. So if you're interested, you can probably in the next few months you could go see that article. But there's some challenges because the data we get is sparse. So we have a lot of zero, and it's also high dimensional. So typically we're going to get, if we do metagenomics, so metagenomic is just we shotgun sequence everything that's in a sample, we're probably going to get like millions of genes. And then we have, best case, we maybe get 100 or maybe 1,000 samples. So this is problematic. There's different method to reduce dimensionality. But I started working with real statistician because I'm not a statistician at all. I'm just playing with the tools. But there's some people that will hopefully help me develop tools that are better adapted for that type of data. And also one point I try to make is there's a trade-off between interpretability and accuracy. So it's very trendy to use these deep learning neural network and things like that, but often it's difficult to understand what's going on inside. It's like a black box. So you're gonna put some input in it, then something happens, and then you get your output. This is very accurate, super accurate, but then it's a black box. And in terms of what we're trying to do, we would rather be able to interpret this model directly or more easy. It's probably possible to posteriori to see what's going on in there, but if you use good old regression methods, you get an equation. Whoops, my equation's gone. So you can get an equation with coefficients that have a sign, and then you can say, okay, this parameter had a positive or a negative influence on my output. And then you can, in our case, we're very interested to go after these parameters. So that's something that's important for us. So I'll show you a first example. I don't know if I'm good, okay. So that's the first time we tried to model the grain baking quality and yields. That was only in 2 fields in Quebec. So in Quebec, I don't know in Saskatchewan if it's like that, but if you ask an agronomist in Quebec, he's gonna tell you like, just put 100 kilogram of nitrogen per hectare, whatever your soil or whatever happens for wheat. So and it's in an official guide for cereal that's what you should do. So people do that and they don't ask any question. And it makes sense if you want to do bread wheat, for instance, you get high nitrogen that's supposed to make high protein, high gluten, then you get this high elasticity, and then you get yummy bread. Unless you're intolerant to gluten, then it's something else. And by the way, I made that bread, so if you're not interested in microbe, we can also talk about bread making. So I started working with a company in Quebec. It's a small milling company, but they're very interested in having high quality flour, and local with. So they want to have the highest gluten and everything, but they were, when I started talking to them, they were like, yeah, this relationship, it's not always happening, you know, there's something else going on, and they were suspecting soil microbes, and I said like, wow, that's my specialty, so maybe we should exchange notes or something. But we started and we did a few projects together, and they were already running a field trial. So this is one field trials they were running. So they were looking at different fertilizer quantities and fractionation over different wheat varieties in Quebec. And what came out of that is that there was no significant effect of fertilization on yields or grain baking quality. And you can see that in the next slide a little bit. We did our modeling and we were able to model it very well. So we have here the predicted yields versus observed yields. And this is for gluten. We did it for all kind of different parameters. And you can see that the relation is not perfect, but if you look at the total nitrogen applied, so I colored the dot with total nitrogen applied, you can see that some plots that were very highly fertilized didn't have very good yield or not very good gluten content, and then some that were not fertilized at high yields or high gluten content. So, and then and it's published, but then it's not, you cannot see it. So, and just believe me, it's been published. So that's the equation we came up with, and that's what we find interesting, is that you can look it up, and you can look here, you can see, okay, there's a negative sign for this near S, which is a gene. I'm going to come back to it. And then there's also a negative sign here with ammonia oxidizer, TRKL1. So if I come back to my nitrogen cycle, I'm going to zoom on it a bit. So the near S gene is the gene that creates the protein related to that. So that's related to denitrification. So more of that gene you have in your soil, the less protein and gluten you add in your grain at the end of the growing season. And these were measured in May, and this is at the end of the growing season, so it's September. So it's like a forecasting tool, we would say. And then if you look at ammonia oxidizer, it's the same thing. More you have of these organisms, the lower yield, lower protein, and lower gluten. And these are the organisms that transform ammonia to nitrate. Okay, you can ask me, like nitrate is good also, it's nitrogen, it's not gone from the system, but if you look at what the plant needs to do if it uptakes nitrate. So nitrates, it's also gonna leach. So the nitrate is negatively charged in the soil also, so generally the nitrate can leach. Also, the plant needs to actively uptake the nitrate, so it takes 20 mole of ATP per mole to uptake nitrate, and it also needs to be reduced to ammonium to create protein. Ammonium is immobilized, so it doesn't move. It's also a passive uptake, only 5 mole of ATP per mole, and it can be used directly for synthesizing amino acids. So we think that if the plant uptakes ammonium, it's less energetically costly than to use nitrate. So we tried to engineer that. So we said, okay, let's go after these, and there's a lot of compounds on the market, like one is N-serve nitropirin to inhibit the nitrification. So these are nitrification inhibitors. And we did that, and it looks like it worked. And then that was very frustrating. The milling company lost our grain, so we could not get the response variable. So we don't know if that affected the grain quality. They just lost it completely. So anyway, we still were able to publish the story, but we had to take another angle. But it's very frustrating, because that was the whole goal, was to have this equation and then see, okay, if it's going to respond in the way we had predicted with our equation. But anyway, I need to move over that. And but there's also, if you look at, like this is pretty, looks like a nasty chemical to me. And we're working with a company that wants to do organic agriculture and like have all these wheat or flour that are organic and everything. And I don't think they're interested in putting that in their fields. But luckily there's a biological nitrification inhibition. So some plants, especially sorghum is well known, and there's other plants that are well known to be able to inhibit nitrification through their root exudates. And there was also one study in Australia that showed that some wheat genotype were able to also inhibit nitrification. So that's a project we started with. So we just grab as many Canadian wheat genotype we could get our hands on, and we're trying to see if they will be able to inhibit nitrification, and then see if that's explaining partly their different quality of grain. So then we tried to expand that. We went to 80 fields. So we started with, luckily I had summer students that just started, and I told them, okay, girls, just get in a car and go all the way around Quebec and take all the wheat seeds sample you can get your, no, it was soy sample. So that was early in the season. So I sent them in May, June to do, basically a road trip of more than 500 kilometers all around Quebec. And with the goal of seeing if we can have also this kind of modeling approach for a larger amount of fields. And this was done by the modeling part was done by Newman, which is a PhD student in the lab. And we were able to create these models with quite a good accuracy. And again, we found that there's some parameters that were quite interesting. So for instance, here you have the fungal bacterial ratio that has a positive relationship with the gluten content. So we tried to go after that. So that's, I kind of, in my nitrogen cycle, I kind of left out some things. I intentionally fooled you, but that was only let's say the inorganic part, but there's also organic nitrogen that's in the organic matter that I didn't put in there, but that is also interesting. So, and this organic nitrogen needs to be degraded by the microbes in the soil. So, and depending on its stoichiometry, so the CN ratio, it will either be released or conserved by the decomposer. And one thing that's pretty interesting is that bacteria and fungi, which are the two main decomposers in a soil, they don't have the same CN ratio. So we think that if the job is mainly done by fungi, they would release more nitrogen for the plant than if it's done by bacteria, because they need less nitrogen per unit of biomass than bacteria. So yeah, that's what I'm saying here. So that was the master project of Emmy, and she did a pot experiment where she was successful to increase the fungal bacterial ratio, and she used 3 different soils, so a forest soil, a field soil, and more a potting soil. And then she also did a field experiment, and it also worked. Whoops, now I just gave away my punch. Okay, but anyway, it also worked in the field, but you can see there's some variation throughout the season. So these are the different weeks of the growing, and it did vary quite a lot, but it was increased. So we were able to modulate this fungal bacterial ratio, and then when we look at the end of the season at the gluten content, for instance, we see that it's in fact negatively related to our ratio early in the season. So the ratio at 2 weeks and 4 weeks was negatively correlated with gluten. So that was another setback. And then when we also had another project that was not necessarily related to that, but we did regression also. That's the project of Newman, and we focused specifically on the field. So that's our experimental field that we have, and we specifically focused on this field, and we sampled it every 2 weeks with the idea of finding the best date to have these predictive models. So we thought, like it's better if it's early in the season, because then you can probably act on what what you understand of your field. But we wanted to prove that, so we did that, and it was indeed early in the season. And if you look at the gluten that was measured on the gluten, that it was measured at the end of the season, but that's with the June 7 microbial data, you see that for that particular field, we still have our ammonia oxidizers, but the fungal bacterial ratio has a negative. So probably this is like an outlier in our regression of our 80 fields, and it's probably one of the fields that doesn't work like the other one, and that's the one we selected for our field experiment. So anyway, part of this work was just accepted this week, so it should be online very soon. So yeah, I'm gonna try to wrap up. So I hope I convinced you that microbes are powerful integrators of physical-chemical parameters, but not only present but also past. And they're also, at the same time, they're key players in many ecosystem processes, so they're very, very important. In our case, our idea is to use this modeling approach to guide us to do the microbiome engineering, so identify the parameters that are interesting and then go after these parameters, try to move them. Could also be used as a modeling, or the modeling could be used as monitoring or diagnosis tool. And again, in that case, I think interpretability is key. There's other methods that we could use. We could have probably better accuracy, but then we would not be able to do what we did with that. So Yeah, so basically microbes are really key, I think, to address many issues facing agriculture. So like the nitrogen use efficiency, like I focus on that, but there's other things that microbes can do, and I think the best would be to really integrate them in either in breeding effort or to understand more how to interact with the plant to make sure that we are able to take the power that they have to modulate the plant phenotype. So with that, I'd like to thank my team. I mostly talk about stuff that Newman did and Emmy, and then Jessica did the sampling, and she stayed for a master and a PhD, so I guess she kind of enjoyed it. And then yeah, we have a website and we have a Twitter, but yeah, and I cannot say I'm very active on Twitter, but you can always try to follow me. So yes, thank you very much again for the invitation.